No mai, haere mai, tauti mai. A very warm welcome from myself, Nicola Batossi. I'm General Manager at Multiple Sclerosis Auckland, and it is my absolute pleasure to welcome you here this evening or this morning. So, Multiple Sclerosis Auckland is a charity we've been supporting people with MS for more than 60 years now and helping them to live full lives. It's our I'm very proud that we are able to provide these information webinars completely free of charge for our community. We provide them on a range of different topics, generally once a month. Tonight's webinar is brought to us through a very special partnership that we've got at the moment with MS Translate, from which we have Brett Drummond joining us. And we're able to bring some really new and exciting ideas and resources to the community. So thank you to everybody who's taken the time to join us tonight. Just briefly, the format will be our speakers will present for about 30 minutes. If you have questions during that time, please feel free to add them to the Q&A box. Some of them may be answered during the course of the presentation. Otherwise, they will be parked until the end. Tonight's talk, as you would have seen, is being recorded, and the recording will be available later on this week through the MS Auckland website. And if you've registered, you'll receive a link for that as well. So without further ado, it's my absolute pleasure to introduce tonight's speakers. Dr. Nick Cunniffe is a clinical lecturer in neurology at the University of Cambridge and a neurologist specialising in multiple sclerosis. He leads the clinical trials and remyelination team in the Cambridge Clinical Multiple Sclerosis Research Group, focusing on clinical trials that aim to repair myelin and prevent long-term disability in MS. His work has helped demonstrate that remyelination is possible in people with MS and is driving the development of new therapies and biomarkers to bring these approaches into clinical practice. Dr. Cunniffe is joined tonight by Brett Drummond, who's a director and founding partner of MS Translate, an internationally recognised MS research science communicator. So without further ado, I will leave you to this wonderful talk. Thank you, everyone.
Thanks, Nicola, and welcome everyone who's joining us from all around the globe, whether or not you're in New Zealand or Australia, or perhaps where Nick and I are in the Northern Hemisphere at the moment, as I've travelled across the world, and I'm currently calling in from London. So Nick, we're in the same time zone for once, but thank you for joining and giving up your time to be part of this. I've had the pleasure of working with Nick now for the past couple of years, and so I know that this is going to be a really exciting, informative webinar. Nick's work in the field of remyelination is really helping drive that forward. But without me talking about who you are, Nick, maybe I know Nicola's given a brief introduction, but can you tell us a little bit about yourself and really what your group is focused on at Cambridge?
Yep. So hello, good morning, good evening, everyone. So I'm Nick, and I'm based in Cambridge. I'm a neurologist by training, as you've heard, specialising in multiple sclerosis. And my main thing is about developing and testing new treatments of people living with MS. And my view is that immune therapies have largely been done in MS, and what we need is a new strategy to try and protect damaged nerves and try and prevent the gradual insidious worsening that some people experience living with MS. And there are various different ways of doing that, but the way we've been trying to get at this problem of progression in MS is trying to harness this natural regenerative process of myelin repair. So that's what I do. That's what my research group does. So I did a PhD about 10 years ago on this project, and have kind of carried the momentum through from that. And I've done two clinical trials now of myelin repair therapies, and also lead a group um, doing research on related structures, testing how best to measure myelin repair, how to capture that in clinical trials. Ultimately, so we can move interesting drugs from laboratory through to clinical trials, and hopefully, in not too long, into patients. So that's what I am and what I'm about.
Yeah, perfect. And so we're going to be talking about a lot of that through the course of the next 25 to 30 minutes. I think one of the things that's interesting that you brought up, and it's a really good place to start, is that this repair mechanism, this myelin repair mechanism or remyelination, is a natural process. And even in people living with MS, we know that this is something that can happen early in disease. But across the course of the disease, that process gets less and less efficient, worse and worse. Do we know a lot about why that process starts to become less effective in people living with MS?
Uh, we certainly have ideas, and certainly we have yeah. There are some factors that I'm very confident are contributory. Then there are others that are less certain. I mean, the key thing, Brett, just to highlight is that so this is the natural response to the loss of myelin. So if you lose the fatty insulation around nerves, what tends to happen is a group of stem cells that we all have. So I was always amazed at the beginning, but 5% of our brain cells are a group of cells called OPCs, oligodendrocyte progenitor cells, OPCs. And those are able to become activated, migrate to areas of damage, make more of themselves, and start to lay down this myelin. And we know that that is the natural response to myelin loss, but it becomes inefficient as we get older. So when you say what factors affect whether myelin repair can occur or not, one of the big factors is age. And there's quite good evidence, both in the laboratory and in people, that your capacity to repair myelin drops off as you get older. Um, but then there are other facets to it as well. The main bottleneck appears to be the activity of the cell. It's usually quite good at getting to areas of damage, but then not so good at the making the myelin cell that it's supposed to make. It gets kind of stuck. So it kind of moves to the area of damage and gets stuck. And there's various different metabolic pathways that have been implicated in that step that we've been trying to target with different drugs. So I think the reason to be optimistic about myelin repair is that it's very effective in people with MS. If you look at a child, often they'll have people with MS who are children who have relapses often remyelinate extensively throughout their brain. And if we can harness that capability into later life, that would be a huge win, because we know that not only does that restore the ability of that nerve fiber to send signals, but crucially, it also insulates and protects that nerve and prevents it from degenerating, prevents it from
Yeah.
the nerve cell from dying.
Yeah. And so the more and more we understand about where those blocks in the process are, the more we can target those. And that's, I guess, where all of these therapies are coming from, is to try and work out, how can we overcome those barriers that exist. For you, if we were to get sort of jumping down to the end of the story, but I think it's important to have that in our heads as we work through this discussion. What would an effective remyelination therapy mean for people living with MS? What do we actually think it will do if we get one of these to a person's life? I know that's not an
Yes,
easy question to answer before we have one, but
no. So the reason why I like remyelination as a strategy is because there's good evidence that it's neuroprotective. So it protects those nerve fibers. So if you leave a nerve fiber that's lost its myelin without myelin for too long, that nerve cell will invariably die off with time.
mhm,
And we believe that underlies some of that worsening that we tend to appreciate with people with MS. Even if they're on the most powerful immune therapies we've got, we still see people gradually worsening with time. So the reason why I like remyelination as a strategy is it's about that insulation, protection, preventing nerve cells from dying off, and in theory, preventing progression,
yeah,
progressive disability in MS. So I think that's um, what I I believe remyelination will do. What I hope remyelination will do is restore some function as well.
Yeah.
So I really hope that a remyelinating strategy, once we find the optimum drug and once we can deploy it in people at the right time, I'm confident that it will have a neuroprotective effect and potentially prevent disability. Whether it can restore function is more optimistic, but a measured response might hope that it would restore a bit of function, but we don't know that.
Yeah. And so I guess we've seen some evidence of this already, and we're going to talk about you've led a recent trial that was the very last talk of ECTRIMS 2025. They made you wait to have to give your big presentation, and we'll talk about that. But there have been trials before that, so I think let's talk about those. What had we seen before CCMR2? What had we seen in the remyelination space in terms of, is this possible?
Yeah, so the best place to start here perhaps is the original story with clemastine, because some people might have heard of that one, but I'll take you through it. So what some researchers, this has nothing to do with our group, nothing to do with Cambridge, but this is some excellent researchers over in the United States. They were studying that step, that differentiation step of that stem cell I mentioned before. And they actually took an interesting approach to it to try and work out which drugs might be effective. But essentially, what they did was they screened a whole library of different drugs. So rather than picking out clemastine because they thought that would work, actually, what they did was went to the end of the process and said, let's get a load of these stem cells in little wells in a lab. And with those with those stem cells in there, and then screen all the different drugs that are licensed at the moment and see which ones actually start to encourage that process of myelin repair in these little plastic wells. And what they found was that clemastine, this antihistamine that hasn't been used for 20 odd years, actually was encouraging these stem cells to start laying down myelin. So what they did was move from that kind of process of screening a whole library of different drugs to identifying a mechanism for how myelin repair is breaking down, and then they put it into a clinical trial. They put the clemastine drug itself, because it's an antihistamine that's already licensed for hay fever, into a clinical trial of 50 people with MS, and they showed that the drugs were associated with an improvement in the conduction along the visual nerves. I think we'd talk about visual evoked potentials later.
Mm-hmm.
But that's the measure they used to capture how fast signals went from eyes to brain, because as you put more myelin on, nerves conduct faster. And they showed that in a clinical trial. So people didn't feel better on the trial, so it didn't have an effect on disability, but they only gave it for 3 months. So that would be very optimistic to see that effect over 3 months. So it's encouraging. It was good evidence that you can promote myelin repair in people. So that's one trial that's been done. The other trial that I'm going to mention is the CCMR1 trial, which is the one that I did about 8, 9 years ago. And that one used another repurposed drug, so a drug that's licensed for another reason. And this drug's called bexarotene. So it's used for a rare type of skin cancer. So unlike clemastine, that used to be widely available, bexarotene is very, very uncommonly used. But this drug targets another block to myelin repair. And we did something similar. We put it into a
Mm-hmm.
clinical trial of people with MS, and we showed that it had an improvement on those eye tests, but also an improvement in the myelin content on the MS lesions, those areas of damage in the brain. So we had kind of converging evidence from the eye tests and from the MRI scan that myelin repair was happening. But again, people didn't feel better over the course of 6 months in that case.
Mm-hmm.
But we knew that myelin repair was happening. So that's where we were
Yep.
up until our most recent trial.
I think it's important to mention just quickly, especially with bexarotene, for people going, well, if we had a trial here that showed that this therapy might be effective at repairing myelin, why is this not going further, there were some safety issues with bexarotene, or some adverse events that were seen with that made it not suitable really to progress further, wasn't there?
Yeah, absolutely. So yeah, I'm very sorry for not emphasizing that before. Thank you for raising that, Brett. So basically, yeah. So while the drug did promote myelin repair, it was associated with several side effects. Some people had rash. Some people had upset stomachs. Uh, yeah, there were some of the side effects were manageable. And I don't think the bexarotene story is completely finished, but we
Hmm.
certainly wouldn't want to use it in the doses that we used it in that trial.
Yeah.
So it might be something we can revisit in the future.
Yeah. Let's move into CCMR2. Before I do, just as a reminder to everyone watching, feel free to drop questions into the Q&A box if you have them. I'm monitoring that, so if we might be able to answer them as we go through, and otherwise we'll do a Q&A at the end, and we'll be able to do those. Let's talk CCMR2, and I think CCMR2 is a really good example showing the entire process that you have to go through to get a drug into clinical trial, or to get drugs into clinical trial, because this was a long story. This morning, as I was preparing for this, I was looking back at a video we did on MS Translate, where I didn't need to look at when the video went up. I could just tell based on me that it was many years ago, based on how young I was looking in the video, talking about some of the initial preclinical work in mice looking at metformin. And I might talk through this little bit to give you a break as well. But just to share through, this was sort of the hopefully everyone can see that. Some of the story here in terms of, and I think Nick brings up a really interesting part of research in terms of the different approaches that can be taken. Going back to the clemistine story, where that was a let's just throw everything at it and see what sticks, versus a more targeted approach where there's something you want to look at. And in terms of metformin, which we'll talk more about in terms of CCMR2, I guess this was more of this targeted approach, knowing what Nick had already said, that one of the biggest issues in terms of stopping this remyelination process was aging. And so this is just sort of highlighting that a little bit here, showing that when we have younger OPCs, and thanks, Nick, for being the one to say oligodendrocyte precursor cell early so that I didn't have to try and spit that out. We see that myelin repair can happen quite well, and as those cells get older, that remyelination happens worse. One of the things that was understood is that one of the ways to sort of combat biological aging was fasting. There was some evidence around that. And so this early preclinical work at the University of Cambridge looked at intermittent fasting in an animal model of MS, where the mice were fed. You had a group of mice that ate normally, and then a group of mice that were only fed every second day, and given disease, and looked at how their myelin repair potential was. And they found that in those mice that were fasting, there was more evidence that remyelination was promoted in those animals. And then wanting to see if that could be mimicked therapeutically, they took metformin, which is a diabetes drug which mimics the process of fasting, repeated that process, and found that mice that were on the metformin also showed improved remyelination potential. And so it was that that sort of then led into the CCMR2 trial, which ended up being a combination of metformin and clemistine. I think two questions, and one that's maybe slightly harder to answer than the other. I know initially we had some discussions with the team, or when CCMR2 was being planned, around why not do a fasting trial? And I think what you told me at the time was, and Will also was talking about this, one of your colleagues, that actually this was done in consultation with people living with MS around Cambridge, and their preference was not fasting. And there's some reasons why in a group of people who suffer from poor energy metabolism and fatigue to start with, that wasn't the approach to take. Do you want to further on any of that, or does that sum up basically
No,
what it is?
just to highlight. So thanks so much, Brett, for articulating that so coherently. That's great. My point, I would come back on the fasting story, is that the rodents, the rats that they are subjected to 6 months of fasting for, it was 3 non-consecutive days in the week, so 3 days per week. And if you scale that up from a rat to a human, because a rat has such a high metabolic rate, actually, we're talking about many days of fasting for a human. You would lose muscle mass. It would be dreadful for people with MS. So that's why the researchers, not in my group, in Robin Franklin's group in Cambridge, that came up with the metformin story, they actually went for metformin because they worked out that it was a way of copying the effect of intermittent fasting.
Mm.
So why subject someone to intermittent fasting when you can do the same thing with metformin?
Yep.
That was the idea. So I'm not sure that whether intermittent fasting might help people with MS. There's no evidence whatsoever that it does, but it can help other elements of people's life, like weight loss, and yeah, there's a literature behind that.
Yep.
But at the moment, for myelin repair, there's no good evidence in people linking fasting to myelin repair, which is why we tried the same thing with metformin.
Yep, perfect. And so the CCMR2 trial ended up being a combination of metformin and
Yeah,
clemastine. Why was that?
yeah. So that's the key process, and it speaks to 2 points we've already made together. So first of all, clemastine um, acts as the signal to those stem cells to tell them to commence repair. But that repair process becomes inefficient as we get old, and metformin can, for the reasons you just presented, can rejuvenate those stem cells so they can respond to instructions from drugs like clemastine. So you
Mhm.
see the two drugs are having a complementary action. The metformin is putting the stem cell in a state where it can respond, and the clemastine acts as the instruction to tell it to respond, to tell it to commence repair. And that's why we use the combination of the two drugs.
Okay, perfect. CCMR2 was in people living with relapsing remitting MS. Just
Yep.
talk to us a little bit about that, because we've had two questions come through in the Q&A. So one from Denise asking whether there are also going to be trials undertaken for PPMS, and I think we might get to that at the end. But just commenting that this was also RRMS. And same, Raymond also asked a question asking if the people on the trials were all relapsing remitting and what level of disability were they?
Yeah. So let's take a step back and say what was the objective of the trial? So we know that these drugs promote myelin repair in animals. We know they're safe to give to people already because they're both licensed for other reasons. Clemastine, an antihistamine for hay fever, metformin's a diabetes drug. So we can bypass the first step of translational research. So the objective of the CCMR2 trial was to see if these drugs that we know work in animals, do they work in humans? Yes or no? Not to work out, is it best to give them to progressive or relapsing? All these different extra questions, which are really good questions, really important questions. So the objective of the trial was to see if the drugs promote myelin repair. Now, intuitively, you would expect a myelin repair drug to be more effective if given early. The reason is that to put myelin back on a nerve, you need to have the nerve there in the first place. Now, when you've been living with progressive MS for many years, you'll still have nerve fibers that are there, that are intact, still functioning, that have lost their myelin. But the proportion of nerve fibers that have lost that substrate, that fiber, will be higher. So you'd expect a remyelinating drug to be still effective, but less effective. So this is all about speed. So what we wanted to do was test if these drugs work, yes or no, as fast as possible. And to do that, we want to do a trial with as small number of people as possible, so we can get the trial done. And we worked out that it was best to test that hypothesis in a smaller number of people with relapsing MS to see if the drugs work. Because if they had no effect at that stage, then I don't think there'd be any point tested. Because CCMR2, and we might come on to, tested the drugs in 70 people with
Mm.
relapsing remitting MS. I'll bet you'd have needed 120, maybe, progressive cases. I
Yep.
think the drugs would have been effective, but less so. So it would have taken us that much longer to get through the trial, if we were recruiting from that cohort.
Yep.
So yes, the answer. So people had lower levels of disability. Yes, the people had relapsing remitting MS, but that's not to say that the drugs won't work in progressive MS, and that is still to be done. But now we've got some good evidence that these drugs are doing something.
Yeah, and I think that's the point, isn't it? As you say, it's to try and generate the evidence as easily we can. It's obviously there's a lot of unmet need in people living with progressive MS, and that's a focus. But we need to be able to get the evidence together to then test it
Yeah.
further. Um, in terms of then the individuals, and you say you got 70 people, and I want to go through explaining your primary outcome measure, which you've already mentioned was VEP. But I think it's important to discuss how difficult it was for you to recruit for this trial, not just because you were trying to recruit through a global pandemic, which made it slightly more difficult. But I think there's an interesting story that's worth mentioning there around how difficult it was to find people to be on the trial and what that means about how far we've come in terms of the treatment of MS.
Mhm. Yeah, so what we've articulated so far is that we're testing two drugs, metformin and clemastine, in people with relapsing remitting MS, and this is what we call a phase two trial. And what we did was test these two drugs against placebos, so drugs that look identical but have no meaningful action, and split the group in half. So I didn't know, and the participants didn't know what they were taking. So that's how to set up your experiment to test if your drugs work or not. And then the question is how to measure it. And as I said before, this is all about speed because I think a remyelinating drug is going to be impactful over the course of years. So I think if everyone took an effective remyelinating drug early, that 10 years later, the chance of, worsening disability would be significantly less. But measuring that over 3 or 6 months would be very, very challenging. So, because it's not long enough.
Mhm.
So what we rely on is tests to measure myelin repair in people, which aren't things like physical examinations, disability assessments. So that's why we use the visual evoked potential as our primary outcome measure, which, excuse me, sorry, is what you just mentioned there, Brett. So some people on the call may have had a VP done before. So if people living with MS may be familiar with this test, um, but it's a method of it's a method of testing the conduction of signals from eye back to brain, and that should usually take about 100 milliseconds, so about a tenth of a second for a signal to get from eye back to brain. But we know it
Yeah.
gets. Oh yeah, you're about to show a video. So this is helpful. Um, just to kind of give you some pictures to work off. Perfect. Yeah. So if you just pause there, so um, so this is what a visual evoked potential looks like. So what we're doing is we're presenting a visual stimulus on a screen there on the left-hand side. So it looks like a chessboard and it flashes twice a second. And then on the right-hand side, you've got a participant who has electrodes placed on the scalp, and we're recording brain waves. And what you can do when you correlate what's happening on the screen with what's happening on the um, EEG, on the brainwave study, is you can measure how long it's taking for signals to get from the eye to the brain. So so Brett's shown me one here. So now what you're looking at here is a electrical recording from the scalp in response to that visual stimulus. And you can see this kind of peak forming in the middle. So it's time along the x-axis. So yeah, Brett's helpfully putting his cursor on there. So that usually takes about a tenth of a second to form that peak. And we know in MS, if you've got loss of myelin, that shifts to the right because it takes longer for that signal to reach the brain. And yeah, this is a helpful animation. So in healthy nerves, it takes about a tenth of a second, whereas in demyelinated nerves, it takes longer. So you can see those signals taking longer to get from the eye to the brain. If you stick myelin back on those nerves, they conduct faster, which I think is another animation you might have in that video, Brett. Um, yeah, so you can kind of see that if someone's on placebo, you'd expect the nerve fibers to conduct more slowly than people that when they're put on the drugs. And that's what we saw. So that's the visual evoked potential and why we tested it. You mentioned about how hard it was to recruit. Well, if you look in the literature back 20 years ago, about 60 or 70% of people living with MS would have an abnormal VEP. So it's quite common to find loss of myelin in the visual nerves. But now it's closer to 35% of people have an abnormal VEP. And that's a reflection of the fact that we're using more immune therapies early in people with MS. So people have less loss of myelin throughout their nervous system. So actually, it was really hard work to recruit people to this trial because not only did you have to have relapsing remitting MS and be enthusiastic about research, but you also had to have an abnormal eye test in the beginning so that we could capture that repair. So we had to, in order to get 70 people, we had to invite 178 people to come to Cambridge to be assessed to see if they were eligible. But again, we did that for speed because we wanted to make sure we were answering this question of were the drugs working, yes or no, and we thought this was the best test to measure that. But we also did other tests. We did MRI scans, we did certain blood tests, various different ways of capturing this process. But we thought this was the best one.
And so I guess getting to the big part of it, what were the results that we saw from CCMR2? Because I think this was exciting when we first heard
Yeah,
about it.
yeah. So we set up this experiment, just like we said. It took us 3 years to get through the trial in total. And what we observed was that people given the drugs had a small improvement in their eye test compared to people that were not given the drugs. So it speaks to that hypothesis we made before that if you have more myelin, you send signals faster, and therefore that it takes less time for the signal to get from eye to brain. And that's what we observed. Actually, what we noticed was that the people that were given the placebos were just gradually worsening that little bit that we tend to see in people with MS. And with metformin and clemastine, it kind of held them at a stable level. And when you compared
Mhm.
the two groups, there was a difference. So that was very positive. It shows that the drugs are having a biological effect to promote myelin repair. Um, but we always must be grounded in what we have achieved. So we've shown that the drugs that work in animals work in humans to promote myelin repair. But again, people didn't feel better over the course of 6 months. If you do a test of disability, we use something called the EDSS as a neurologist, which is when neurologists start tapping people with hammers, getting them to walk, getting them to hop, testing their strength, their sensation, things like that. It forms a score of their disability. And we compared the disability scores in the drugs compared to the placebos. There was no effect. But I am not disheartened by that at all for the reasons that I
Mhm.
mentioned. Because actually, if you are living with relapsing remitting MS, if you are on a good immune therapy, because most people are nowadays, in that early stage with relapsing remitting MS, you're worsening very slowly,
Mhm.
because the drugs are so good now. So in order to show a protective effect of a remyelinating drug against that gradual worsening, you either need a really powerful remyelinating drug, which I don't think these drugs are. I think they have a small effect, but I don't think they're very powerful at remyelination. Or you have to go long. So to see
Mhm.
that difference in the two groups, you probably have to go 5 years down the road. But I was really encouraged by the results because it showed that what we're doing in the animals, translating it through to people, it is having an effect to promote myelin repair.
Yep. Interesting that you talk about, obviously a lot of people at this point are on pretty effective therapies. One of the questions that we received in the Q&A box was around the trial and did participants have to go off their DMTs to be on the trial? So
No,
very easy to answer to that was no.
no. So everyone had to be on a disease modifying treatment because we wanted the immune part of MS to be controlled so that we can study the repair without that noisy element of ongoing damage. If that makes sense. We want people's MS to be stable and then to capture the repair on top of that.
Yeah, perfect. I want to finish by talking about where we go next, and there's definitely some questions that tie in with that. We'll then have a little bit of time for some extra Q&A, so definitely put any last questions into the chat as well. One of the questions that I think is a really interesting thing just to bring up before we move to the next steps, Shelley's asked, does the new myelin, after it gets repaired, still get attacked? Or can it still be attacked in the future? I think it's an important question.
Yes, it can, which just speaks to the point we just made about the importance of being on some sort of immune therapy as well, so we can suppress the immune component of the MS as well. So so yes, in theory, it remains vulnerable, and ultimately it kind of speaks to what our vision is for people with MS in terms of treatment. So
Yep.
the analogy I often use is look at something like HIV, the virus, HIV virus. Now it's very much a disorder where people are very controlled because as soon as you get diagnosed, you have three treatments. You have a triple therapy in terms of medications. And I would like MS to be the same. I would like people to have almost three therapies. I would like an immune therapy, another therapy to kind of protect the underlying axon, something called neuroprotection, and I'd like a remyelinating therapy as well.
Yep.
So ultimately, this is what you need. You need these three different treatments. We've got good immune therapies now, and there's still some work to be done in this area, particularly in progressive MS, but we're making some progress there. But then what we also might need to review is the neuroprotection strategy and the remyelination strategy.
Perfect. You just answered a question that came through from Eric, so good job there. If we talk about next steps, I think that's obviously where people want to know where this is going. And we'll get into a little bit of detail in terms of what you think, what are the next trials? What do we need to do around this? But let's start with the big question. Steve asked it. It was the first question that came through to the Q&A. What sort of time frame are we looking at to get an effective therapy? What's the best guess, knowing that there is no real answer to this?
So the first thing I will say is that I don't think metformin and clemastine in combination are the solution. I think what we've got here is good evidence that these drugs promote some myelin repair, but it's not anywhere near as effective as I was hoping for. But also, but I think the learnings from this is enormous.
Yep.
So what's really, really important and valuable is that what we've been able to achieve so far is with drugs that are licensed for other reasons. What we now want are drugs that are specifically tailored to this myelin repair strategy. And the most excitement I've got about this area is actually that industry, so pharmaceutical companies, are now getting really excited about remyelination.
Yep.
And they wouldn't do that if they didn't think it had a future.
Yep.
I'm having more and more conversations with companies that are developing new drugs specifically tailored to target mechanisms that are important for promoting repair. And crucially, they'll be able to do it more effectively, and in theory also without the same side effects as well. And that's where I think the future lies. I think what we've been able to achieve is demonstrate this as possible, and I think there's a bit more work for the academic side to drive this forward.
Yep.
To make we're better trialing, etc. But ultimately, I think what's going to happen is we're going to end up with a novel myelin repair drug that's going to come through the whole pipeline. But in truth, how long is it going to be? Maybe 5 or 10 years.
Yep.
I'd be confident that I'm prescribing a remyelination drug in 10 years. And I'd be hopeful I'd be prescribing one in 5 years. Now, the final thing to say on that note is this is kind of where we were with immune therapy 30 years ago. So in the 90s, there was just interferon, And
Hmm.
then 20 years ago, then there was Tysabri, and then over the last 10 years now, all of a sudden we've got 20 licensed drugs. I kind of feel like we're just in the early 90s for myelin repair, and we're probably going to get one drug in the next 5 years, but then hopefully in 20 years we'll have 20 drugs
Yep.
promoting myelin repair. So I'm optimistic about it.
Yeah, I mean, I think we're at you've highlighted it really well. We're at a really exciting stage with this, but there's still work to do. I think the fact that now there's been 3 trials that have shown this is possible in people living with MS is a huge step forward. We know that we have that proof of concept, and we're at a point now as well, not least to do with work that, you've significantly contributed to, of knowing how to conduct these trials, which is a really important thing as we start to get better, more and better compounds to know, we can put them in and test them appropriately. On that topic, a question from D coming through. Is clemastine going to be progressed as an option? Do you think there's any future for clemastine in the remyelination space, or do you just think it's not effective enough?
So I think a drug like clemastine might be important. And
Okay.
what I mean by that is that I don't think clemastine gets into the brain in sufficient quantities to really boost the repair enough. So what I think is a drug will come along that targets the same mechanism that clemastine does, but does it more potently. So um, at the moment, if one of my patients comes into my normal NHS clinic and they say, "Should I be taking clemastine?" I say no, because people get tired on it's a sedating antihistamine. People feel tired on it,
Mhm.
um, and we haven't got the good evidence that the drugs are actually having that long-term effect. And I'm not sure clemastine on its own is enough. So the reason why I'm optimistic about it as a treatment is not clemastine itself, but because I think it's accessing an important pathway that we might be able to
Yep.
do better with newer drugs.
I think that does bring up a really important point that you and I have discussed before, and we've said is really important messaging to get out about this. People talk, and I hear it through MS Translate a lot. People are able to access some of these compounds because they are available, can access metformin, can access clemastine. Um, I think it's important that there's messaging around. There is nothing from the data that we have at the moment, from the trials that have come out, to suggest people living with MS should be taking
Yeah.
these medications, that they're going to see any sort of benefit from it. Do you want to sort of comment
Yeah,
on that quickly?
I completely agree. Like I say, if I thought these drugs were the solution, I'd be telling people to go and get them themselves. But
Yep.
I just don't think we're there yet. But that doesn't mean I'm not optimistic about this as a treatment strategy for people with MS. It's
Yep.
just that we don't have good evidence that these drugs are having that long-term effect.
Yep.
But yeah, these are kind of
Yeah. No, I mean, I think I completely
steps
agree. They're a step
steps
along the
along the
way.
path. But it's important because lots of people living with MS are very sensitive to the literature and keeping
Hmm.
a presumably your attendees here are all people that are interested in MS research, which is why you've all logged on today. And yeah, I think if I was living with MS, I would also have similar questions and
Hmm.
think, oh, well, I've just heard about this interesting area of research. Should I be getting the hold of this drug myself? Because metformin is widely available.
Yep.
But at the moment, I don't think that's the right thing to do
Yep.
at all for MS because we don't have the evidence that it works.
Perfect. A few more questions come through, and we've got Nick for a little bit longer. Um, Lisa has asked, I think, we've covered this briefly, but let's address it specifically. Do you know if there is a possibility for nerve remyelination for people who have lived with a diagnosis of MS for 20 odd years without much visible disease progression? Also considering those in their 60s and older. Do you want to do you
Yes,
want to tackle that one?
so to come at that point, yes, I think there's a possibility for remyelination to occur. Um, in CCMR2, for instance, the average disease duration was 13 years. So these are
Mhm.
people that have been living with MS for quite a while because there's a range around that. So some people had had MS for 20, 30 years on the trial, and we didn't see a clear effect between duration of disease and the effect of the drugs. So I'm optimistic, but it comes back to that point I made before. I just think the drugs will be less effective when you've been living with MS for many, many years, but not ineffective.
Yeah.
So but that's one of the reasons why we deployed metformin, because we thought it would make the drugs more effective in older individuals.
Yeah.
People on CCMR, the final point, sorry, on that, CCMR2 only included people from 25 up to the age of 50. And we didn't actually show that the combination was any more effective than clemastine on its own.
Yeah.
So whether metformin's added anything, I don't know. Um, I think it probably has, but I don't know that. And what we may need to do to appreciate the effect of metformin is actually give it to older people,
Yeah.
to people sort of 50 to 70, because maybe that's the people that need the metformin to rejuvenate those stem cells so they can then have the positive clemastine effect. Maybe if you're 30, you only need clemastine on its own, and maybe if you're 60, you need metformin and clemastine. So
Yeah.
that's just what I mean. That's why we went for that rejuvenating effect. So yeah, I'm optimistic, but I just think ultimately remyelination drugs are going to be more effective given early, but I still think they can be effective given late.
Yeah, and I think, I mean, you talked before about where we are with the immune modulating therapies. Um, and I think what we've seen with those is that the importance of having so many different options is that we know that different people respond differently to the different medications. And so you need to find the one that works for each individual. And I think, my personal opinion, and be good to hear your thoughts as well, is that there's no reason to suggest the same won't be true for remyelination, that we may just need to find the right one for the right person at the right time.
Yeah, yeah, I think so. But ultimately, it speaks to that point about having that axon there, the
Mhm.
nerve fiber there ready to repair.
Yeah,
And yeah, so I think, yeah, I think I said my bit on that. The key thing is that I think the drugs will have an effect, just yeah,
yeah,
less, but I'm still optimistic.
yeah. Question from Denise that's just come through, I guess specifically around your group. What are the plans for next steps in terms of trials in this space?
Yeah. So, um,
Still
so
recovering from CCMR2 probably, so you need a little bit of
well,
downtime from
I've
what
been
was a
trying to
huge
get
trial.
I've been trying to get funding for the next one for the last 2 years.
Hmm.
And the problem is that there is limited resource for researchers nowadays to fund these big trials. We've been very lucky in the UK because the MS Society in the UK are incredibly supportive of us in Cambridge. So they've been the main funder for CCMR2. So, but doing these trials is not cheap. So
Hmm.
I've got my ideas as to what I want to do for CCMR3. But I've been trying to get that funded for the last 2 years and I haven't been able. And they need about £2 million. So
Yeah,
yeah, it's a big ask, but
process.
I'm still trying. So
Yeah,
an awful lot of what I do is write grants trying to get these things funded.
yeah,
Um, so yeah, so that I would like to do CCMR3 because I think I know what I want to try next. The other thing to say is that point I made earlier about industry, about
hmm,
pharmaceutical companies that are coming along with new drugs, and they want their drugs tested properly, and therefore are starting to speak to people like us and the group in the United States, and there's some other groups in Australia as well. We're working on this area, and I'm quite excited about some of the new drugs coming through. So I want to be part of that too. So
yeah,
so from the trial point of view, there's a CCMR3 that I want to get funded, and I'm also hoping to support the industry effort because my motivation here is just to get these drugs through to test if they're effective and if they are effective to get them into people with MS as soon as possible.
yeah,
And those are the two ways forward I think that we've got
yeah.
for making that happen.
Okay. So if anyone out there that's watching might have a spare £2 million, they
Look
should
behind the sofa.
they should reach out. Christine's asked, going back to CCMR2, um, any notable results seen from the MRI
Yeah, so
side of things?
yeah, so the main MRI result that we had showed that there was an effect on MS lesions, but it was very small, and it was different depending on how damaged the area was in the beginning. So what we saw was that if the lesion, that ball of of inflammation on the scan if it was badly damaged at the start, didn't respond so well. So what you want is kind of this Goldilocks zone of, a bit of damage, not too much damage, just right in the middle. And those ones seem to respond a little bit. But it wasn't, an amazing, very, very positive result. It just showed us actually that not all MS lesions are built equally, and some are more damaged than others, and some will respond more than others.
Hmm.
So that was the main lesson from the MRI. But it was helpful to have that support from MRI alongside the eye tests and some other tests that we had as well, all pointing in the same direction and showing the drugs had had an effect.
Yep. We've had a couple of questions come in, one from Jackie and then one from an anonymous attendee, just around non-pharmacological approaches to
Yeah.
this and whether there's any evidence that they might um, promote remyelination. Any thoughts on that? I mean, I know that there's been some studies that have shown exercise can
Yeah,
promote
yeah, that is the main
BDNF.
thing. That is
Yeah.
the main thing. So there's no diet that's been shown to promote myelin repair, but actually I'd quite like to test that too.
Hmm.
But I don't need 2 million for that, but I might need a little bit more money. But I would like to test diet in myelin repair. But exercise is a big thing.
Yeah.
And there is good evidence in animals that if a nerve fiber has more information going down it, so more activity, that can actually help support that repair process. So exercise is important, I think, in myelin repair, but no one's tested that in people yet. But there are other reasons to be active and to exercise when you're living with MS. So
Yeah.
again, if people came to my clinic and said to me, should I be exercising to try and promote myelin repair? I'd say there's lots of different reasons why you should be exercising. So please do. Don't go
Yeah.
mad, but physical activity is really important, within the scope of what you can manage.
Yeah.
So go on, Brett. So that's my point on exercise.
No, I agree. I mean, I think there's been some stuff that's shown that it can promote some neuroprotective compounds, BDNF being one. But yeah, I think there's lots of reasons why we should all be doing exercise, and yeah, also people living with MS in particular. Look, I know we've got only a minute or two left, so I've got two questions to ask you.
Mhm.
Um, we'll just do some quick fire answers to those, and then I'll thank you for being here. Scott has asked, do we have a view as to why an antihistamine like clemistine might be useful in the treatment of MS?
Yeah, so it's not because it's an antihistamine,
Yep.
is the key thing to say there. So clemastine is actually doing multiple things, and the reason why it affects myelin repair is because it has an off-target effect on another pathway. So it's not because it's an antihistamine. So if you
Yep.
go away and take loratadine or Piriton or one of those drugs that you might see in the shops for hay fever, they won't affect myelin repair.
Yep.
So it's just clemastine that has this off-target effect.
Perfect. And the last question before we let you go, question around GLP-1 medications and have they
Hmm.
shown any potential around remyelination?
People are looking into this. So
Yep.
remyelination is a bit of a up in the air question. Some people think it might do something, others not. But there's a possible neuroprotective effect of GLP-1 drugs outside of myelin repair.
Yep.
Uh, these drugs have already been tested in things like Parkinson's disease, and showed a small effect on mobility. So there are people who are advocating GLP-1 drugs be tested in MS, and I have no doubt that that's going to happen soon.
Yep.
Um, but how big an effect they have, I'm not so sure.
I mean, it's a question for now. All right. Well, there's a few more questions. I can hang on a bit longer, but I might take this opportunity, Nick, to say thank you. And I don't know, Nicola, if you want to also come back on, we'll let Nick um, leave us. But thank you so much for sharing all of that, Nick. It's been super.
No, it's been lovely to speak to you all, and take care. Thank you so much for having me.
So, over Nicola, anything that you want to say? I'm happy to continue to answer a few questions if there's some more coming through. I think we answered most of them. But otherwise, I'll hand back over to you.
Just to add from my perspective, it was an incredible talk, and it's really highlighted for me just how complex this area is and just how many kind of factors there are involved, and quite interesting to look at the progress in remyelination against the sort of time frame of the immunotherapy treatments, with that sort of ideal utopia of the three-pronged strategy sometime in the future.
Yeah. And I think, the fact that a lot of these different approaches that we're looking at for remyelination are coming at it from different angles, that adds a lot of positivity around it, because it means that you've got a lot of shots on goal. And if one approach doesn't work, others are already being progressed that may. So, as Nick said, I think we're at a really exciting point in this, and the fact that, someone who is very much leading the charge on this says very hopeful that he'll be able to prescribe this in 5 years, but very confident that within 10 it'll happen. I think that's a pretty exciting thing to be able to hear.
Absolutely.
So I'm just looking through just to see if there's any more questions. So just a couple of quick answers that I can provide. So Christine asking about current medications and whether they'll help with remyelination. None of our existing therapies have really shown potential to repair any of the damage. They don't work in that way, but they slow new damage from occurring. So certainly very much worth taking from that perspective. The more we can slow damage from occurring, the better it is. And hopefully then, as Nick was saying, we'll be able to add in remyelination therapies in the future that then can start to work on top of that to repair. And then Trisha's question around had MS for the past 24 years, problems with mobility, but not in a wheelchair, confirm what the benefit would be for a remyelination therapy. Certainly not on an individual level. At this point, we don't know what it would be. I guess as Nick said, just to confirm some of those points. We're hopeful that remyelination therapies will be found that can benefit people, regardless of how long they've been living with MS. People on the CCMR2 trial. The median time frame of disease duration was 13 years. So, we're able to push down, and there are things around what might be needed to help people who have been living with MS for longer. So, certainly a lot of hope in there. And I think that's all of the questions. Let me just. So there's another question's just come in about metformin and the person having access to metformin and whether they should try it. So first thing around that is always recommend talking to a healthcare professional before doing anything, but to really emphasize the messages that Nick stated as part of this. The benefits observed on CCMR2, while it was positive from a scientific perspective, the actual benefits to people living with MS weren't really observed. They didn't feel any better being on these therapies, and there were side effects associated with it. So metformin, known in people living with diabetes as well, and we're seen in the trial, can cause some gastrointestinal problems. Lots of people had upset stomachs, diarrhea. So there's, it's certainly nothing at a point, as Nick said, metformin and clemistine are not at a point where we would be recommending any person living with MS is taking them. There's no evidence to suggest that should be something people could do, despite the fact that we obviously understand that there's that thought around it. I think that's everything.
Lovely. All right. Thank you so very much, Brett, and obviously to Nick as well for tonight's session, which was just really, really fabulous to hear exactly where we are right on the yeah, right on the front of the research world. So thank you so very much, and I hope you have a lovely day there in London.
Thank you.
And to the rest of everybody here tonight in New Zealand, Pō Mārie, thank you for joining us.